Hey everybody, it's Mr. Fabeck here. We are talking about phabiology and mutations today. So it's our favorite topic because of all those movies like the X-Men and Marvel's universe, the Hulk, Spider-Man, all of those characters, and my own personal favorite growing up, the Teenage Mutant Ninja Turtles. We're going to talk about how those happen and what was going on with their DNA. So by the end of the presentation, you're going to be able to explain 3 different ways mutations happen in cells and explain how mutations affect the individual. You know you mastered this if you can identify the type of mutation when comparing DNA strands and explain how this mutation affects the amino acid sequence. And so you even further know that you're exceeding this mastery if you can tie it into other subjects such as meiosis, genetics, and evolution and ecology. So look at these, okay? So we got some crazy things going on there in nature. We have this duck that has extra feet, this turtle that has extra heads, snakes, toes, Raphael. These are all mutations that somehow happened and developed in our body. So a mutation, by definition, is just a change in the DNA that affects genetic information. And that genetic information is represented as proteins. And so all the stuff we're seeing is the protein code that is essentially affected. So when we change our blueprint of our DNA, we're going to change the transcription message of mRNA, which changes how we translate that into the protein code. So here we go. Looking at this, we have 2 strands of DNA. One is original, one has been mutated. Take a glance at that. What do you notice is different? As you're looking, hopefully you noticed that this section right here, just one letter changed, and so now we have mutated DNA. So how does that affect the individual? We're not quite sure at this point. But what about if we had something like this? Now we have 2 different strands, and again, we're looking at the C and the G also changed. So what's happening? What's going on? What's going to happen to our individuals? Will I finally get the X-ray vision that I've always wanted, or the laser beams, or maybe just those sweet ninja skills that I wanted as a child? So what is a mutation? We're changing nucleotides, right? So we're changing our DNA, what we talked about just before. And so we have 2 new terms that we haven't really mentioned before: somatic versus gametes. So gametes we're familiar with meiosis. We're making sperm and egg cells. They have half the number of chromosomes, and when they fuse together through fertilization, we're passing that genetic or those genes to our offspring. Whereas somatic cells are all the cells that are not sperm and egg cells. So these are things that we don't pass to our offspring. These are cells like your hair cells, skin cells, nerve cells, muscle cells. These are things that are part of you. So if we get a mutation in a somatic cell, it only affects the individual. If we get a mutation that's in our gamete cell, that's going to affect our offspring. So we can think about this whole idea of mutations and genetic variation as we talk about ecology. And so we can think about one mutation in an individual that leads to having new offspring with that mutation can lead to changes over time and then changes in the entire population, whether it's beneficial or harmful for that species. So is it harmful, helpful? Well, sometimes we don't really see it, so we could probably say a lot of mutations are pretty much neutral. They have no impact, and we can think about that with our codon chart. If you look at your codon chart, there's multiple ways to do proline and glycine. There's multiple ways we code for that same amino acid. So changing one letter for some of these may not have any effect because we'll still bring in the exact same amino acid. Whereas if we change one letter that maybe causes it to stop early or puts in a new amino acid, well, that's how our proteins can be made wrong, and that's how we can have these mutations. So some example of some neutral ones: we have eye color, birthmarks. These are things like freckles that happen over time or maybe just spontaneously happen that only affect that person. Does this person have perfect eyesight? I personally don't know her, but maybe she does, maybe it doesn't. But either way, it's not affecting their offspring and not affecting them directly. Same thing with freckles. It's really only affecting that person, and I can't really think of a harmful aspect of a birthmark or freckles other than to watch your skin in case it turns into skin cancer. But for the most part, it doesn't affect our daily life. Harmful ones, if we want to think about like skin cancer and all the different forms, as you can see, these are somatic forms, right? And so we have different variations of skin cancer that are affecting that individual and those cells there. And we can also think of food allergies. Sometimes they say kids grow out of their peanut allergies. Sometimes food allergies develop as we go on, right? Like this idea of gluten and this not being able to digest wheat products. So these individuals most likely ate wheat for their entire life, but then all of a sudden something happened and now they're no longer able to make the protein that's digesting those components. And so food allergies are a little bit more relevant, and they're hard to diagnose, but they're most often caused by some sort of mutation. Beneficial are rare. I mean, you got to think about where you are in the world, right? So that's the idea of evolution ecology. If you got a mutation where instead of being blue or purple, now you're green and you can blend into the environment, well, that's a beneficial mutation. But if you got that purple color and now you're in a white environment, like the snow or something, you're going to stand out. So that's not beneficial at all. But here's an example of a real life one that could be considered beneficial. Sickle cell anemia. So we can see our picture here of sickle cell anemia. We see our red blood cells that are normal here. We see our crescent or sickle cell blood cells, and those sickle cell blood cells do not carry oxygen as well, and they're not as flexible, so they can get clogged up in your arteries and cause problems with strokes, heart attacks, and just general blood flow. Now you should be saying, Fabiology, Mr. Fabeck, that doesn't make any sense, right? Why could this thing that causes strokes and heart attacks and blood problems be beneficial? Well, if we look at the map of where we find higher prevalence of sickle cell anemia, there's a higher percentage of African populations with sickle cell anemia, and ironically, we see a similar map with the amount of malaria. And we know malaria is a blood disease caused by a protozoan, and that is affecting healthy red blood cells. So if this sickle cell is a codominant trait where some of your blood is normal and some of it's the sickle shape, people who have the sickle shape are not getting those cells affected with malaria. And so they're still able to do some functions, so they have a better chance at surviving, whereas somebody who has healthy red blood cells has more blood cells for the parasite to really attack, and so therefore those people may not survive as much. And so we're seeing a higher prevalence of sickle cell anemia in African Americans, and we can see there's a relationship that helped develop over time between malaria and those individuals. So let's talk about the different types. We're going to talk about point mutations, thinking about substitutions, replacing one letter, insertions, and deletions. So a point mutation is where we're changing a single nucleotide. So one letter that we saw earlier today, G to C, getting switched anywhere in our DNA code. So a substitution, just like if you had a substitute teacher, is you're replacing one nucleotide. So I like to think of it as if the ribosome's reading a message, this is the message that would be read. So instead of the fat cat ate the rat, maybe the ribosome reads the fat hat ate the rat. So it doesn't quite make as much sense. So it's not the exact same message, so it's not the exact same protein. And we can infer that if all of these words are representing an amino acid, we have a new amino acid for whatever this hat amino acid would be. So we changed one amino acid in the code, which could cause some sort of problem. Maybe it's that food allergy, maybe it's that freckle. Frame shift mutations, frame shift, these are the ones that are going to mess you up. These ones mess you up a lot because what you're doing is you're inserting or adding nucleotides, one or more, or you're deleting nucleotides. So it shifts the entire reading frame. That's why we call it a frame shift. If you think about how you read a book, you're just like a ribosome reading. You're reading your codons in groups of 3. So deleting a letter or adding a letter shifts the groups of 3 off, and so that changes the whole sentence. So ultimately, this changes how your proteins are built, and if they're not built properly, if it's an enzyme, they're not going to digest that food properly or digest or break down or put together things that they normally would be activating. So these ones are the most harmful. So an example, we added nucleotides, maybe we add in a whole new amino acid. Again, that can change the shape of the protein and not have it function anymore. We could delete or take away. So again, we could have the fat ate the rat, and that's completely taking away an amino acid that we would need for some sort of function or product. But here's where we get the big frame shift. Adding one letter, like this H, shifts everything down, and so therefore we have 1, 2, 3, 4 different amino acids that are different than what we started with. And I want you to notice that there's an extra letter at the end. This can help you identify which one you're looking at. Same thing with deletion. I just delete one letter. We have 1, 2, 3, 4, 5 different amino acids that are different here. So again, everything's shifted because you still read your codons in groups of 3. So here's some example problems that I think that you should check out. Compare this original DNA code to your 3 here. A little helpful hint, the easiest thing is insertion deletion. Just count up how many base pairs you have in each one. So if you count how many base pairs you have here, and you count up the base pairs in your mutation, if they don't equal the same, you can tell if you added or subtract. Then go back letter by letter to see what's changed. Okay, so this is your mastery page. This is what's going to help you out. Thank you for watching, and if you have any questions, always feel free to stop on by. Thank you. Bye.